It's really our pleasure to invite again somebody who you've seen in the street of head of the Verbana unit of IRSA, but now he will be a speaker. And then the name is again Emanuele Ferrari. He didn't change his name since yesterday. He's a biotechnologist specialized in proteomics, metaproteomics, and in the study of melanocytes. He's working with bacteria, fungi, but also mammals, applying his expertise in different ecological topics. And again, since October 2024, he's also the head of the unit of IRSA. So it's our pleasure to have Emanuele Ferrari. We should have clapped the hands also for Esther, but yeah, later.
I will come with that. Like,
okay.
Non lo so, si vede lui che parla? A lei?
Sì.
Okay. So, hi again. So we have seen something on genomics, but now we are taking to the next step, which is the thing that is actually interesting, that is proteomics. Yes, I will convince you that it's much better than genomics. I'm joking. The two techniques are very connected, and they go together very well. And I will show you that actually, if you have both, you will have much more information. So, it was working like 2 minutes. Okay. So this is a bit of what we are gonna see this morning. So what is proteomics? How you want to do your experimental design and sample preparation, which is something, if you want to collaborate with the proteomic expert, you have to do this part. And then I want to show why proteomics is useful. And in the end, we have a little bit of metaproteomics, which is a bit different because we have different questions. And as Esther pointed out before, you need to have a question before starting to this kind of analysis, because it's very easy to say, we will analyze a lot of samples, and then we have a lot of proteins, and then in the end you cannot make sense of those data because you don't have a question in mind before doing the experiment. So proteomics is obviously the study of proteins, which is the end product of these things that you study in biochemistry 101. And it's not the study of a single protein, of a bunch of proteins, but a whole set of proteins that are expressed in a certain condition in a certain time. And you want to look at those proteins in an unbiased way, because you are not selecting the proteins, but you are identifying and quantifying all the proteins in that condition. So this is more unbiased. You still have biases always because of instruments and the type of technique they are using, but it's not a bias that you introduce that you want to select some of the protein. And you can study whole organism, especially if you have small animals, bacteria, fungi, but then you can also go on subcellular organelles. If you want to study just the membrane proteins, you can do it. It's a bit tricky, but you can do it. And then something especially interesting for fungi is the study of secreted proteins, since they have a lot of extracellular metabolism, I guess, you tell me. And this is what is fancy called the secretome. And then now you can also do on single cells. It's still developing technique, but we are there, I'd say. It's it's harder than doing genomics on a single cell, but you can do proteomics now. And then metaproteomics, which is the study of microbial communities. So why proteomics? Why choose proteomics over the other techniques? The first thing is that from one gene you can have more than one protein, especially in eukaryotes. So if you are characterizing the genomes, you don't know everything that happens when the cells translate the proteins. And the other things that I think it's the main advantage is that proteins are dynamic. So they change with condition, with time, depending on the strain that you have, etc. So what you are going to see with proteomics is what the cell is doing in that particular time, and that you cannot do this with genomics. You can say that also we can use transcriptomics. Yes, it's similar, but in the end, not all the mRNA is translated. So there are changes that you cannot see with transcriptomics. And there is no overlap between the transcriptome and the proteome, basically. So there you can use both technique, and you will not have a complete overlap, but you can look at the function that you see. So with the two techniques. Obviously, if you have an infinite amount of money, you can do everything, and you will have a lot of data, and then you can integrate all this data. And then one thing that you may know is that proteins are modified with post-translation, and this can change the function of proteins. So if you want to look at this, for example, phosphorylation changes a lot the metabolism of cells. So this you cannot. This is the only. You can only see this with proteomics. And in the end, you also have metabolomics, which is just the study of secondary metabolites, which is something also interesting, but I think it's the hardest thing to do, especially for some organisms. So in conclusion, genes are, they say, the receipts of the cell, but in the end, it's the proteins are like the functional players, and they are the phenotypes of the So first, you need to have a question. What we usually do in proteomics is have different conditions, and we want to compare them, because it's not an absolute quantification with proteomics. So you have to have in mind that you need to compare different things. Maybe you have one strain that you want to, for in this case, they wanted to see how protein expression change with different raw media, or you can you can grow them with different temperatures. You can change the condition and see what changes in the metabolism. So which proteins are expressed and what is changing. So this is the thing to have in mind when you want to do proteomics. So we have, first of all, this very briefly, you want to you have to isolate to extract the protein, and then you have a digestion. So you are not looking at the whole protein, but at peptides. Okay, just to have an idea of what you are doing, and then the main technique is mass spectrometry, which is the gold standard for proteomics. But now there is also some nanopore protein sequencing that is becoming much interesting, and I think it's very less expensive because for a mass spectrometry, we are talking, if you want to do it in your lab, it's like 500,000 euros, basically. And with a nanopore sequencing, I think with maybe 50,000 euros, something like that, you can have something, but it is still a developing technique. But what we do also here is to have services for proteomics that now are growing. Like, it's not like genomics or transcriptomics, but you have a lot of services now, and you can have like, for example, will be like from 2,200 euros to 500 euros, more or less, for sample. So it's a bit expensive, but not so much. And I think it will decrease with time. And so for the sample preparation, what we have to take to consider that there is no PCR for proteins. So what your sample, the proteins have to be there. You cannot amplify them. So you need more sample usually. But now with the instrument sensitivity that is increasing, this is becoming less of a problem. As I said, now they can look also at single cells. But in the end, you need a good amount of proteins. It's hard to say how much because it depends a lot on the samples, and basically you have to rely on your experience, on the literature to see how much you need. And you have also to consider that different samples, it means different preparation, because if you have a fungal cell, it's different from bacteria. You have the cell wall, etc. So, and sometimes you have to do a harsher condition to retrieve the proteins. And also, especially with proteomics, metaproteomics, sorry, you need to consider that some materials can interfere with the analysis. For example, the typical example is the soil metaproteomics. You have a lot of humic acid, something, and other things that you need to remove. So in the end, you have also to consider this. But now there are also for this, there are a lot of kits that are designed for this kind of analysis. It's becoming much more simple. This is the workflow for the analysis. This is one of these mass spectrometry. I will not get into detail, but what I want to just clarify that in the end, you have this raw data that are a list of mass spectra. So something that is not understandable, and you need to assign for each spectra a peptide. So that's we're doing. So this is the workflow. So as I said, the first thing is to identify with the protein and quantify them. Then you have quality control, and then you have a comparison of the different conditions that you used. And in the end, you want to know which function are your proteins have. So the other thing that you have to consider is that you need a database of proteins. So if you're studying a very strange bacteria that no one ever looked into, you will not have a sequence of proteins already in your database, and that could be a problem. Because it's much easier, and the standard is to look at these two databases. One is UniProt and the other is CBI, and see, okay, is my organism there? Someone has sequenced it, someone has put the list of protein there, and that's what we usually use to identify proteins. So other question when you're planning to do proteomics experience, okay, let's look into UniProt or CBI if we have already a proteome there. Otherwise, there is another things that you can do that also Esther talked for. You can go from the whole genome sequencing, from transcriptomic data, and build your custom database, which is very nice from an analytical point of view, because you have a very good data your sequence, but in the end you need to do this, and it's not super easy. Okay. Otherwise, there is this technique that is de novo analysis that doesn't have any in common with the de novo sequencing, but it's bit tricky. And in the end, what you have is a list of protein like this. So you have a name, a code, a name, a gene name usually, and then you have a list of condition and a quantification. And for this, you can have like now for a good experiment, you will have like 10,000 proteins for each sample, more or less. So if you receive from your proteomic analysis a list of 10,000 proteins like this, I think you cannot understand anything. So what we want to do is to have like
What I'm sorry, on the previous slide, I didn't understand what are the conditions. Maybe I missed it.
No, it's just I put some. There are like this is from actually a tumor cell lines. It was
What conditions could be experimental treatments or?
yeah. For in this case, there are 3 different cell lines, and one is untreated, and the other is treated with radiation, for example. So you can compare or different strains, for example, or conditions. So one is treated, the other one is untreated, and you can have a pairwise comparison or multiple comparison. And then what you do, the first thing you do usually is to have like this comparison between the conditions. So they could be, sorry. You can have a simple t-test, which is the normal thing to do. So if you have only 2 conditions, you do that. Otherwise, you can do an ANOVA. If you do the t-test, now the things that they do is usually you have this volcano plot. So you have some proteins that are overexpressed in one condition, and other proteins that are overexpressed in another condition. So you can see the difference between these 2 conditions. But also in case now you have this, but you don't know what are these proteins doing, So which is the biological significance of this data. And also you can do if you have different, in this case there are different strains, for example, of medicinal mushroom. You can do this cluster analysis, so you can see which strains are similar. So, for example, this one has similar protein expression, and this one are similar, and this is very different. Okay, so with this you can have an idea of what are the differences between all your samples or all of your conditions. But also in this case now, these are all things. These are like 5,000 proteins for each sample, but you still don't have any information. But from this, you can do what is called function analysis. So you assign to your proteins a function. And what I always say that you are not looking at the single proteins, but at the functions globally. So we are not saying, okay, I want to look at the specific protein, because otherwise we will get crazy, looking at 10,000 proteins, but you group them for functions. And for doing this, you have this like gene ontology database. Yeah, I forgot what also here you need to have some database that tell this protein is doing that. So each protein will have like a tag saying this is for nitrogen metabolism, this is for carbon metabolism, etc. So you need some information, okay? And one of the main databases that is used is this gene ontology, which is quite, there are a lot of information on every different kind of organism, and they can give you where the protein usually is expressed. So nucleus, membrane, extracellular, what the protein is doing, like is it phosphorylase or other kind of specific function, and in which metabolic pathway is involved. And usually this one is the thing that one is more interested into, and what we usually look at. So the biological process in which your protein are involved. And I think this is what proteomics is much more interesting. And another thing you can do is like study the network of proteins. So how the proteins in your sample interact. And also for this, there are some databases that can tell you which are the interactions. And this is very useful, especially because sometimes you have, you know that some proteins interact, but you don't know in this interaction, there are some proteins that have known function, and other that are unknown. And so with this, you can also assign a possible function to this. For example, here you can see a small network of proteins, but obviously these are all related. And if you see something like this, you can say, okay, in my sample there are a lot of keratin in this case, so they can have a function. Could be important for my sample in those conditions, and these are histones, I think. So with this kind of visualization, you can look at the cluster that are much more enriched, and so which function are more important. This is just there are some repository where there are a lot of data, but if you want to play with proteomics and do some analysis just for training, you can do it. And that's it on proteomics. And now I will briefly say something about metaproteomics, which is a bit different. And I think for some environmental studies is much more interesting. And the difference is basically that you are not looking at a single organism or 2 or 3 organisms, but a whole microbial community. Okay, so for example, you can have the study of soil metaproteomics, sea, lake metaproteomics, and the interesting thing here is that you can link the taxonomy to the function. So you are not just saying there are these bacteria there, but you know that the bacteria 1 is doing this, the bacteria 2 is doing that. So it's basically you are assigning a function to each one. So with metagenomic, you can say who's there, so what is the potential. But with metaproteomics, you say what they are doing and what they are their metabolism. In this case, I have to say for this kind of metaproteomics, you need metagenomics, because the easiest thing to do is to at least an identification of your community and say who's there, and then you can do the metaproteomics. And so the difference is that you have very bigger database with respect to proteomics, a lot of species, and so you need much more computational power to these things. They're very huge databases. It's a bit tricky to do that. But now there are a lot of different tools that can do this. And the workflow is basically the same. You have an expression table, so a list of proteins, a list of taxa. You can do the differential analysis also here, even if the statistic is a bit tricky, I have to say. You can assign function to each of proteins, see the interactions, see the metabolic pathways, see also the clustering. But the difference is here. What you don't have in proteomics is that you can do a taxonomic analysis. So see in your sample which kind of taxa are there and what they are doing. So this is the main goal of basically of metaproteomics. This is an example of the things that you can see. So this is just an identification, and then these are the taxa associated to each function. I don't know. I don't like this kind of plot, but it's not always clear for me. Okay. Or also you can have something like this, more like this is just to see who is there. Okay. Instead with this you can see what their metabolism. And yeah, this is an example they did it with the on the ocean, Arctic Ocean, I guess, and they assign different function to different taxa. For example, they saw how the carbon acquisition change, the nitrogen acquisition change, and which metabolism are differentially expressed, let's say. And taking account then for this kind of analysis, you need some liters of of water, seawater, to do something like this. There was an old, I think the first one started with 300 liters, but I guess it was 10 years ago. And now maybe, no, I think you can do it with 10 or 20 on the concentration. With the sensitivity increase of the instruments, now it's becoming much easier to do. Also with the lake, I think you can do it with 10 liters maybe, but it depends a lot on how much bacteria you have there, basically. Okay, so if you want to know something about metaproteomics, there is this metaproteomics initiative, which we are part of, and they have a lot of tools for very basic for every kind of level of knowledge. And they have a lot of paper there, videos, very nice. And also, if you want to, this is a bit of self-advertising because we have this paper that will possibly come out this year, and now there is only the preprint of the archive. But if you are interested in the microbiology, in the omics applied to microbiology, sorry, this is this network which is Microgalaxy, which is now trying to promote this tool that are on this Galaxy server. Maybe some of you learned, has already heard about this. And on this website, you can see a lot of tools from metagenomics, metaproteomics, metatranscriptomics. There is a lot of these things, and registering on Galaxy will have a space which is 250 gigabytes, I remember, that you can load your data there, and you can also use this server for doing the analysis. So you are not using your computer. With a very basic computer, you just have to load your data and then do all the analysis on the server. So it's not a huge space, but in the end, few analysis is very useful. And there are a lot of tutorials and tools there. It's super easy. And if you want to join, there is also a possibility to join the Microgalaxy community. There are and you can also contribute to these tools. And on the preprint, you can see all these things well presented. And finally, what we can do here as a group, as I said, that we are not doing the instrumental analysis, but we are now developing protocols for sampling preparation, and also we are now developing protocols for the analysis of data, which is not always easy because it depends a lot on your organism. So if you are interested in this, you can contact me and we will chat later. And I will just leave this as a summary, but I already told everything before. And so this is what proteomics and metaproteomics can do for you. Okay.
Thank you very much, Emanuele. I'm checking whether there are questions from home, and I give you 5 minutes, let's say, for question here in the hall, in the room, and then we launch the coffee break. Any question?
Thank you, Emanuele. This is really interesting. I was wondering, and this is because I have absolutely no idea. When this last example on the metaproteomics, for instance, that you said that usually you couple the metaproteomics with the genomes.
Yeah.
So we know that DNA is quite stable and stays in the environment for a little bit of time, but I have no idea about proteins. So how can you be sure that these proteins are coming from live and active organisms, or are they just proteins that are there, but maybe the organisms are not there anymore? So how long the protein stays? Couple of weeks, and how Can you trust that these are really coming from these?
Yeah. It's hard to tell how long they stay, but if you saw them, usually they are from live organism because the majority, I would say 99% of the proteins are from live organisms. So what you're seeing is like a picture of that specific moment. What you have to take into account if you take the sample at the morning in the sea, it's not the same as in the evening, for example. That's a bit, so it's a picture of that particular time. But in the end, there are, I think, I would say 99.9% of the proteins are from active cell. So because the organism doesn't produce proteins if they don't need it. So if you have, it's because they're using.
Also for those that are excreted.
For the secreted, it depends a lot on the type of sample because if you're looking at water, they can be everywhere. But for example, in biofilm, you can see because they are secreted, but in a closed environment. No? So and there you can separate the secrete from the intracellular.
You can also use the growth moment with it,
Yeah,
higher for people.
If you're doing culture, obviously it's easy to see which are separated.
It's like what we did, we grow up Leventus fungus that we filter the fungus from the broth, and then we analyze the broth and the biomass. So you can have an idea of intracellular.
Yeah.
Even if it's not pure.
Thank you.
I have a related question. You answered it partially already. If proteomics or metaproteomics is so sensitive to the conditions, like you may take sample in the morning, you put it on the machine or fix it in the afternoon. So the sampling conditions and your handling or the experimental treatments that you maybe have in the lab, and how you agriculture there for the experiment, and you want to measure these proteomics from these experiments or the treatments. What you need to consider to not have artifacts from sampling or from experimental conditions. How can you fix it? Or
there are some in the lab you can freeze it. Just put it in the minor 80s, it's fine. It doesn't change so quickly. And I think for environmental sample, there are now some reagents that you can put to block. It's not like for fixation or alcohol fixation suitable, but I know the name, but there are some key. And I told about this thing that from morning to and evening differences, because I saw a presentation at this metaproteomics symposium, and they were demonstrating this in the Baltic Sea. It was completely different, the hour of the day and the kind of composition.
and in the lab, you would need to think about how you have the controls so that you can compare it always to the controls, because the lab setup might.
You need a baseline, for example, so you have basic condition, and you test your what you want to know, for example, the effect of some contaminants to your organism. But then basically what you do is a subtraction. You subtract the baseline from your condition.
I just had one announcement. Sorry.
For example, if I have samples from biofuels from a lake, and I want to check if the proteins that I have in APS or in this biofilm are coming from actually bacteria or from other sources. Metaproteomics answer to that?
Yes.
It's a question.
If you can distinguish where the proteins in a biofilm, for example, can come, are coming, or from bacteria or another organism. And yes, unless you can, I have to say there are some limitations if you want to distinguish with two strains of the same bacteria. Sometimes the proteins are the same, obviously, or the sequence is the same. So in the case no, but for example, for different genera have different proteins, so you can distinguish that. And if you want to say from bacteria or fungi, there are completely different sequences, and so yes, you can distinguish.